Welcome to our poster on learning the score of conservation at the functional genomics level for human and mouse genomes. Our work is motivated by the fact that it is important to understand the differences between human and mouse given the wide use of mouse model in biomedical research. Given the increasing size and diversity of functional genomics data from both species, we reasoned that it would be beneficial to develop a method that learns meaningful information from a large collection of human and mouse functional genomic annotations. We therefore developed LeCIF, which stands for Learning Evidence of Conservation from Integrated Functional Genomic Annotations. As shown in this figure on the left, LeCIF leverages large-scale functional genomics data from human and mouse to quantify evidence of conservation at the functional genomics level. We call the genome-wide score learned by LeCIF score, as shown in the bottom of this figure. One notable feature of LeCIF is that it does not require the user to explicitly match experiments from different species based on their data type or biological source, which allows us to leverage data from a diverse set of cell types and assays. In other words, you can include an experiment or an annotation even if it exists in only one of the species. LeCIF trains an ensemble of neural networks where aligning human and mouse regions are considered positive pairs, and the same set of regions that are randomly paired up so that they do not align to each other at the sequence level are used as negative pairs. Each positive or negative pair consists of two feature vectors, one containing functional genomic features annotating the human region of the pair, and the other containing features that annotate the mouse region of the pair. In training, LeCIF weighs negative pairs 50 times more than positive pairs, which makes the resulting LeCIF score distribution right-skewed. This makes it extremely rare for a pair of human and mouse regions to score high, which allows us to highlight regions with strong evidence of conservation. As features, we use p-calls from DNA-seq, histomark and transcription factor chip-seq, and cap analysis gene expression experiments, and additionally chromatin and chromatin state annotations and RNA-seq signal. We note that sequence alignment is only used to define positive and negative pairs and not used as input features. Feature labels such as DNA-seq and human brain are also not provided to the classifier, and we make predictions for every aligning pair of human and mouse regions. In the bottom here, we show what the score looks like in the genome browser and highlight certain high scoring and low scoring pairs as examples. Here on the bottom, we also demonstrate that LeCIF, which uses an ensemble of neural networks, is better at predicting aligning pairs or positive pairs than baseline methods such as random forest, deep canonical correlation analysis, and logistic regression. Using features provided as input to LeCIF and also external annotations, we validate that the score reflects similarity in functional genomic signals as expected. For example, transcription start sites and promoters score high, whereas enhancers score relatively low, reflecting the well-established notion that enhancers evolve faster than promoters. We also find that LeCIF can provide complementary information to sequence constraint scores, which quantify conservation at the sequence level. We lastly apply LeCIF score to external cross-species data sets relevant to diabetes, and find that the score is able to highlight regions that exhibit similar phenotypic properties between human and mouse. Overall, we believe that LeCIF will be a resource for mouse model studies. More information on this work can be found in our publication, which is shown here in the bottom right corner. Thank you.